molecule that can bind specifically to a TCR or antibody. Let me here talk about TCR in summary. PCR and PRR. What do you know about them?
TCR, T cell receptor.
This is T cell receptor. Yes.
B cell.
And this one is B cell receptor or indeed membrane bound antibody. B cell receptor is indeed membrane bound antibody. And this pattern recognition receptor for innate immune cells. So there are antigenic receptors of the immune system. We have different types of receptor in the immune system, including cytokine receptors, chemokine receptors, growth factor receptors. But now we are talking about antigen receptors. Okay, antigen receptors. We have three main types of antigen receptors in immune system. Indeed, by using those receptors, immune system can sense the antigen, can see the antigen. They are the tools of immune system that by which immune cells can see the antigen to check if it is foreign or self-antigen, normal or abnormal antigen. But TCR is for T cells, BCR for B cells, and different kind of PRRs, including TLRs. Do you remember toll-like receptors? I'll talk about them. PRRs, they are antigen receptors of innate immune system. Okay? So they are antigen receptors. So any substance or any molecule that can specifically bind to TCR and antibody is called antigen. Okay? It's called antigen. But immunogen is any substance or molecule that can generate immune response. Gen is from generation of immune response. Immunogeneration indeed. So any molecule or any substance that can generate immune response, that can induce activation of T cells and B cells is called immunogen. So all antigens are not an immunogen. All antigens are not an immunogen, but all immunogens are antigen. Is that clear? Yes. All immunogens, because they have to bind to TCR and BCR and induce immune response. But all antigens cannot induce immune response. For example, they are self-antigens. They are normal antigens. Self-antigens do not activate T cells and B cells, so they are not immunogen in normal circumstances. So what is the PRR binds to what type of antigens? They are bind to antigen. I told you anything that binds to BCR and TCR is called antigen. But for PRR, pathogen associated molecular patterns and damage associated molecular patterns. Down. So immune cells can sense both pathogens and damaged cells, indeed abnormal self-antigens. It's better to say now another term is used instead of PAM because for example LPS, lipopolysaccharide, is not just for pathogens. It is expressed by all gram-negative bacteria, both pathogen and non-pathogen. So the better term is microorganism associated molecular patterns. Molecular patterns. This is M. Instead of PAM, it is called MAMP, microorganism, instead of pathogen. They are the same term. They are the different terms for same molecules. Clear? So, what What is toleration? What is tolerance?
Select the normal cell from the abnormal cell.
Toleration of
system.
self-antigen by immune system means tolerance. Toleration of self-antigen means tolerance, or indeed having no immune response. Being indifferent against self-antigen is called tolerance, and any antigen that induces tolerance is called toleration. And most self-antigens are indeed toleration. Okay? No question? They are just definition. What is Hapten?
Small molecular.
Very small molecules
Can't have any.
that they can bind to the TCRs, but they cannot induce immune system by itself. It has to bind to a carrier protein. It has to bind to a carrier protein in order to induce immune response. For example, penicillin is a small chemical antibiotic. Penicillin is very small chemical that by itself cannot induce immune system. But if the penicillin binds the RBCs as a carrier, now it can induce immune response. That you know that some people have allergic reaction for penicillin or unwanted reaction to penicillin. It means that penicillin has bound to the RBCs and it has induced immune response. Most allergic reaction to drugs or medications, most allergic reaction to drugs are indeed Hapten carrier effect. It means that most drugs are small or tiny molecules that they cannot activate T cells or B cells by themselves, but when they bind to a protein, including RBC proteins, then they cannot induce immune response. So what is epitope or antigenic determinant? There are different terms for one thing.
Part of the protein.
A part of a big antigen. For example, I told you this table is a big antigen, but TCR and this, for example, this one is TCR. TCR cannot recognize, bind to all of them. It can bind only just a small part of them. This small part is called epitope, and it's expectedly a big antigen is possible to have several epitopes. Both same epitopes or different epitopes. Is that clear? When we have a big antigen, it's possible to have different or same epitopes. And having different few numbers of one epitope on the antigen is called polyvalency or multivalency. So polyvalent antigen means that one specific antigen that it has several of one specific epitope, not just one of that epitope. If an antigen has several epitopes, same epitope, it is called multivalent. Okay, it's just terminology. So we have two types of epitopes: linear determinants or linear epitopes and conformational epitopes. What's the difference between them? Linear and conformational.
Maybe it depends on the chain of the amino acids.
Sequence.
Sequence, yes.
Yes. This one, for example, we have 10 to 12 amino acids on the chain that one TCR or BCR can bind to it. So we have a normal sequence of amino acids on the protein. This is called linear epitope.
GCL can recognize the linear peptide of the protein on the MHC, but antibody or B-cell receptor can recognize
Conformational and linear, both of them.
yes.
Thank you. So we have linear, that these linear antigens can activate both T cells and B cells, but conformational, for example, if different parts of the chain that they are come together because of the conformational change and they make an epitope. This is called conformational epitope. If a protein is denatured, then we won't have the conformational determinant. If we lost the conformational epitope. Is that clear? That's why this conformational epitope can activate antibody only, not T cells. Because for T cell activation, we need antigen processing and cutting and presentation on the cleft of MHC that I'll talk about it. And this MHC peptide only can activate T cells. So when we have just peptide, so there is no conformational epitopes cannot activate T cells because conformational epitopes cannot bind to MHC. Only linear epitopes bind to MHC. And another type of epitope is called neoantigen or neoepitope. It's as a result of changes on the sequence of, as a result of mutation or cutting by enzymes of a protein. When we have proteolysis or cutting of a protein by enzyme, we have a new epitope. That this new epitope wasn't in the normal protein. So as the name shows, neo is indeed new antigen. That we have epitopes that it was absent in the normal protein. But after cutting with enzyme or after induction of mutation or any other chemical changes, we will have new epitopes. And this kind of new epitopes can activate T cells and B cells. That's enough. So, factors influencing immunogenicity. We can categorize them in different groups. First of all, I'll talk about the factors that are contributed to the immunogen itself. So, we are talking about the properties of an immunogen. What properties of an immunogen can increase the immunogenicity of an antigen? When we say we have two antigens with different immunogenicity, for example, antigen epitope or antigen A and antigen B. Antigen A is more immunogen than antigen B. What does that mean? More immunogenicity, what does it mean?
Or less immunological response to be activated.
Yes. What else? What else? When we say we have an antigen which is more immunogenic than the other one.
A weak signal can activate it, can stimulate it.
Can
A weak response, immunological response, can stimulate it.
Can stimulate the stronger immune response.
So, yes.
Yes, yes, that's right. So when we say we have an antigen with higher immunogenicity, it means that that antigen can induce a stronger immune response. When we say an antigen have lower immunogenicity, it cannot induce a strong immune response. So some properties of the immunogen can affect the immunogenicity of an antigen. The properties of the immunogen. For example, foreignness. What does that mean? Foreignness of an antigen can influence the immunogenicity of an antigen.
This antigen is not normal.
No. Foreignness. Foreign antigens versus self-antigens. When we have self-antigen or normal self-antigen, they are not immunogen at all.
Yes.
But the antigens of the for example, microorganisms, they are immunogen, the stronger immunogen. Is that clear? So we have autoantigen, alloantigen, and xenoantigen. What does that mean? Autoantigen are the antigens of the person owns a body. Self-antigen indeed. So self-antigen is autoantigen. And in normal situation, we won't have any immune response against self or autoantigen. Okay? But alloantigen are the antigens of one human to another one. It is important in transplantation or blood transfusion. So, and the xenoantigens is, in clinic, we are not dealing with xenoantigens. Xenoantigen is mainly, we are dealing with in the laboratory, research laboratory, not in the clinic. But from the concept of foreignness is important. for example, antigens of a human A and human B and rabbit. So the antigens of human A for B for human A and rabbit for human A and the antigens of itself, which one is more immunogenic? The antigens of rabbit, it means if we inject, for example, a tissue, blood of, for example, blood of rabbit to human, or another human to human, or the blood of himself, which one is more immunogenic? Rabbit, because it is foreign, and when it is foreign, immune system can recognize many epitopes on it. When immune system can recognize many different epitopes on it, it can induce very strong immune response. But when we transfuse blood from human to another human, we have some similarities, but they are not exactly same. So we will have some immune response, but not as strong as this one. But the antigens of the blood of one person to himself or herself is not immunogen for it. It means this. So foreignness is very important in immunogenicity, because if the antigen is more foreign, it will have more different epitopes from the host that is receiving the antigen. Clear? Another factor is size of the antigen. The antigens more than 10 to 14 kilodaltons are the most immunogenic, and very small antigens, for example, Haptens, they are very small that by themselves they alone are not immunogen. But some small antigens are immunogen, but the larger antigens are more immunogen. Why does it so? That the larger antigens are more immunogen compared to smaller antigens.
Easy to be detected, easier to be detected.
How? What do you mean? They can be, yes. One reason is that they can be captured by macrophages easily when they are larger. The small proteins cannot captured by macrophage, phagocytosed by macrophages easily. One reason is that. Another reason is that the bigger antigen, the higher chance of having different epitopes. When we have different epitopes on the antigen, we have the higher chance of to have, indeed, when we have different antigens, we have activation of more T cells. When we have a lower number of epitopes, then we have activation of lower number of T cells. Okay? So the larger antigen, it is possible that have more epitopes, and when we have more epitopes, we will have activation of more T cells. When we have activation of more T cells, it means a stronger immune response or a stronger immunogenicity. Another factor is chemical complexity. The more complex antigen, the more immunogenicity we'll have. The simple antigens are not a potent immunogenic antigen. For example, amino acids are not immunogen at all. But complex proteins, heterodimer proteins, or heterotetramer, or multimer proteins are more immunogen. You can see here that, for example, proteins are more immunogen than polysaccharides, and the polysaccharides are more immunogen than nucleic acids and then lipids. And you know that only proteins can activate T cells. I'll talk about this. Polysaccharide and nucleic acid and lipids, they can only activate B cells. Why just protein can activate T cells? Why polysaccharide, nucleic acid, and lipids cannot activate T cells? What's the reason for that? Why non-protein antigens cannot activate T cells?
Because the type of the antigen that's loaded on the MHC molecule is just a peptide protein.
Yes, just proteins and peptides derived from them can bind to MHC. But polysaccharide, nucleic acid, and lipids cannot bind to MHC. When any antigen cannot bind to antigen, it cannot activate T cells. So when we have a protein antigen, this protein antigen can activate both B cells and D cells, both of them. So we will have a stronger immune response. But a polysaccharide antigen can activate only B cells. So we have a B cell response without any help from T helpers. It means this. So polysaccharide, nucleic acid, and lipids are the antigens that they cannot activate T cells. So we will have B cell response without receiving any help from T cells. So when we do not have help from T cells, the immune response would be slower or weaker than the proteins. Okay? No question? And usually the more complex or more complexity again, if a protein is more complex than another protein, when we have more complexity, indeed we have a higher chance again to have different epitopes. More complexity means having a higher chance of having different epitopes. Again, when we have different epitopes, we will have activation of more T cells, and it means again a stronger immune response and a stronger immunogenicity. Another factor is stability or degradability of an antigen. It means for protein antigens, especially for protein antigens, that they are going to bind to MHC and molecules. This complex protein usually is cut by enzymes to produce small peptides. So peptides derived from this complex protein binds to MHC. Production of those peptides is done by a complex of enzymes. So if we have a protein, a complex protein, this protein cannot bind to MHC. It's too large. It should be cut by enzyme to small pieces, which we call them peptides. Now these peptides can bind to MHC. So if a protein cannot be cut by enzyme, it cannot be bind to MHC. Degradability means this. A protein should be degraded by the enzyme to small pieces in order to be able to bind to MHC and to activate T cells. When a protein cannot become degraded by enzymes, it cannot bind to MHC. We need peptide to bind to MHC. If the protein cannot be cut with the enzyme, it's very, its degradability is low. When the degradability of a protein is low, indeed its immunogenicity is lower than the other. Question, comment? So, they are the factors influencing the immunogenicity, that all of them are contributed to the nature of the immunogen, the properties of the immunogen itself. But physical form is it's very similar to the size and complex. Let me skip this one. So, factors contribution to the host. When we say immunogenicity, it means that we have a host that is receiving an antigen. So, this antigen should be immunogen in order to induce immune response in the host. Okay? I told you that it should be foreign, it should be protein, it's, for example, degradable, the larger one, all of them will increase the immunogenicity. Indeed, all of them will increase the chance of having immune response against this. But we have some other factors in the host that indeed affect the immunogenicity or immune response against this immunogen. Some factors contributed to the host that receive the antigen. Okay? For example, genetic factors. You know that, for example, you saw COVID-19. Some people recovered from COVID-19 without any symptom. Some of them had flu-like, cold-like symptoms. Some of them had flu-like symptoms. Some of them were hospitalized. Some of them were died of COVID. So different peoples with different genetics had different response to COVID. Is that clear? So genetic factors is important to induction of immune response against any immunogen. For example, if we have a same immunogen, same antigen, if we inject this antigen to different people, we might have different immune response against them. Because the immunogenicity of the antigen is stable, but the genetic of the hosts or people receiving this antigen is different. So when we talk about the genetic factors, what factors we are talking about regarding the immune response, immunogenicity? What factors or genetic factors inside our body can influence the immune response against an immunogen or antigen? What factors? You have the slides.
Can you repeat the question?
What kind of genetic factors can influence the immune response of a person to an immunogen, to an antigen? What genetic factors can influence the immune response?
Species of the
No. We are talking about genetic of, for example, human to human. We are not talking about different species. For example, I told you, for example, we have a vaccine that we injected the vaccine to different people of the persons of, for example, different for you. Immune response induced by you is not same. Some of you may induce a stronger immune response, some of them may induce weaker immune response. And it's because of genetic factors. So what genetic factors can induce immune response?
Immune system.
Yes.
Yes.
Genetic factors related to immune system, any genes of immune system. For example, HLA, I'll talk about it. HLA is very important in induction of immune system. 2% might have different HLAs. So as they have different HLAs, they will induce different level of immune response. Or receptors, antigenic receptors, TCRs, BCRs, PRRs, or any other antigens. We can put cytokines, cytokine receptors, antigen processing, antigen presentation, phagocytosis, genes that are involved in phagocytosis, genes that are involved in NK cell activity, genes that are involved in T cells and B cells. Hundreds of genes are involved in all immune responses. And any variation in those kind of genes can affect the induction of immune response. Another factor is the status and physiology of the host. What does that mean? For example, age. People with different age will induce different immune response.
Yes.
Very young and elderly individuals usually cannot induce a strong immune response, because in very young we have immature immune system, and elderly we have declined immune system. Another factor is immune competence. Some people might receive some therapeutics, some medications that, for example, chemotherapy. That chemotherapy, you know, that suppress immune system. So if a person receives immune suppressant drugs, like chemotherapy, it can induce immunogenicity. And disease and inflammation, if the person have now a disease or not. If the person have disease or inflammation, it can affect the immunogenicity. And another factor is prior exposure. If a person has previous exposure to the antigen, of course it will have memory cells, so it will induce a stronger immune response because of the memory cells, you know. Another factor influencing immunogenicity is method of administration. Route of administration indeed. When you can see here, we can inject the antigen in subcutaneous, intramuscular, intradermal, intraperitoneal, intravenous, and oral or mucosal. And you can see here the ranking of the routes by their immunogenicity. If the antigen is injected in subcutaneous, it can induce a stronger immune response compared to, for example, oral administration of the antigen. So the subcutaneous administration of antigen from subcutaneous will induce a stronger compared to the other, and when we come down, the immunogenicity will decrease. Another important, very important function is dose of antigen. Antigens in very low dose and very high dose cannot induce a strong immune. Usually the optimal dose, middle doses of each antigen is the optimized or the best dose for the induction of immune system. So very low dose or very high dose of antigen can induce tolerance. Another factor is adjuvants. What is adjuvants? Have you heard about it anything? When we inject antigen vaccine, other than the antigens of the vaccine, for example, when we are developing vaccine for COVID-19, we need to put antigens of COVID inside the vaccine. Besides the antigens of COVID, we have some other products that increase the immunogenicity of the antigen. They are called adjuvants. Adjuvants with different mechanisms, I'm not going into the details, with different mechanisms, mainly by activating of innate immune cells, by engaging of different PRRs, they can induce a stronger innate immunity, they can induce a stronger inflammation, by then they can induce a stronger adaptive immunity. So adjuvants are chemical or biological agents that are produced together, that are injected with the main antigen in the vaccines in order to induce the immunogenicity of the main antigens. I'm not going to go to the details. So we have one types of antigens are T independent. We have T independent antigens and T dependent antigens. What is the difference between them? T independent antigens and T dependent antigens. What is the difference between them? I told you in the previous slides. T dependent antigens are protein antigens.
Yes, and this is
Because protein antigens can activate T cells and B cells as well. But non-protein can only activate B cells. They are called T independent antigens. Polysaccharide, nucleic acid, lipids, they are non-proteins. They are T independent. But proteins, any macromolecule that have protein, they are T dependent. That's, you can see here, for example, that's enough. That's enough. Knowing the definition is enough. T dependent antigens, they are proteins. Example, microbial proteins. So you know that some microbes have only a polysaccharide capsule. For example, pneumococcus. Do you remember? Pneumococcus has a polysaccharide capsule, so it can only induce a B cell response. If I talked about Haptens, for example, here is the example of Haptens. Penicillin by itself cannot induce immune response. Then it binds to the carrier, a protein or RBC, proteins of the RBC. Now it can induce immune response. So there is a difference between the antigens that are detected by TCR and BCR. The difference between antigens that are detected that are recognized by TCR and BCR or B cells. So all kind of macromolecules can activate B cells, including proteins, polysaccharides, nucleic acids, and both linear and conformational antigens, both of them can activate B cells. So it doesn't matter that the antigen is protein, polysaccharide, lipid, nucleic acid, or they have linear or conformational. All of them can activate B cells. And in terms of size, it doesn't matter. For T cells, just proteins. Non-proteins cannot activate T cells. And just linear determinants, not conformational determinants, can activate T cells. It is the main difference between, and for T cells, we must have the peptide bind to MHC. Without MHC, we won't have any T cell activation. So for T cell activation, just proteins and just linear epitopes, and of course we need MHC molecules. That's enough. What is superantigen? Superantigens are some products of microorganisms that they can induce very strong immune response. They can induce a huge number of T cells, and they can even induce anaphylactic shock that I'll talk about them in the next slide or next lectures. So I'm not going to the details now. For example, the conventional antigens can activate just this cell, but the super antigen can activate several cells, can activate a huge number of T cells. When we have activation of a huge number of T cells, we will have a huge amount or huge level, a very high level of cytokines, inflammatory cytokines that can induce anaphylactic shock that I'm gonna talk about it in the next lectures. I talked about the determinant by the innate immune system, PAMs and DAMs, and they are detected by PRRs. That's enough for this lecture. 10 minutes break and get back to the. Please get back in 10 minutes.